All right, let's jump right in. We are living through what might just be one of the most incredible moments in the history of medicine. We're talking about a fundamental shift in how we even think about disease, moving away from just managing symptoms to actually rewriting the source code of life itself.
And yeah, this quote, it really hits the nail on the head. For decades, medicine has been about managing things, right? Keeping chronic conditions in check. But now, we're on a totally new frontier. We're talking about editing the very source of genetic suffering. It's a mind-blowing change in what's possible.
So you might be asking, how is this even happening? Well, it all comes down to the difference between ex vivo and in vivo. The first wave of these therapies, like the cure for sickle cell, was ex vivo. You take a patient's cells out, edit them in a lab, and then put them back in. Pretty cool. But the real revolution, the in vivo approach, is like sending a microscopic surgeon directly inside the patient's body to fix faulty cells right where they live.
Okay, so we've got this molecular scalpel. But what is it really? And maybe more importantly, how does it know where to go? How does it not just, start cutting up everything in sight? Well, that brings us to the incredible technology making it all happen.
You've probably heard of it, CRISPR Cas9. And that term, genomic surgery, is absolutely perfect. This is, without a doubt, the most precise tool we have ever had for this job. You can basically think of it as a find and replace function, just like in a word processor, but for our actual DNA.
And the way it works is just brilliant in its simplicity. It's a two-part system. First, you've got the guide RNA. Think of this as the GPS. Its only job is to hunt down and locate one very specific faulty gene. Once it locks on target, step 2 kicks in, the Cas9 protein. This is the scissors part of the system. It follows the guide and makes a perfect, precise cut or edit. So elegant, yet so incredibly powerful.
So that's the theory. But let's look at how this works in the real world. One of the most exciting battlegrounds for this technology is in the fight against cystic fibrosis, a disease that has stumped scientists for decades.
And this is where we hit a major hurdle. See, with a blood disorder, you can take blood out of the body. But lung cells, you can't just scoop them out and put them back. They're buried deep inside you, protected by layers of mucus and all your body's other defenses. So the big question was, how in the world do you get the CRISPR machinery in there to do its job?
For doctors and researchers in pulmonary medicine, solving this has been the absolute holy grail. The goal is to get in there and directly correct one specific genetic typo, the delta F508 mutation, right in the lung cells where it causes all the damage. Not a treatment, not a patch, but a permanent fix.
And this is where the solution gets so incredibly clever. Scientists have basically created tiny biological delivery drones. They package the CRISPR system inside either little bubbles of fat, called lipid nanoparticles, or inside harmless re-engineered viruses. And the best part? These can be turned into an aerosol. That means a patient could, theoretically, just breathe the cure in from an inhaler, letting it sneak right past the body's barriers and get to work. It's just brilliant.
Now, making the edit is one thing, but making sure it's safe, making sure it's effective, that is a whole other challenge. And that part of the story is all about data. A massive, massive amount of data.
Just look at that number, terabytes. That's the amount of data we can get from just one single patient in one of these trials. To put that into perspective, your phone probably has a few hundred gigabytes. This is thousands of times more. And making sense of that data mess is absolutely crucial for these cures to succeed. And this is where the unsung heroes come in, bioinformatics and AI. They create the bridge from a cool idea in a lab to a real working therapy. Using these powerful AI tools, scientists can actually model a patient's entire genome and predict what will happen when they make an edit. They can catch potential off-target effects, mistakes, basically, before a single drop of medicine ever touches the patient. It just goes to show you, the future of medicine is as much about data science as it is about biology.
So the science is it's mind-blowing. But of course, a technology this powerful doesn't exist in a bubble. As these cures become reality, they force us to ask some really big, really tough questions as a society.
And the first question is usually about money. We are talking about potential price tags of $2 million, maybe even more, for a one-time cure. So, how can our healthcare systems possibly handle that? Who gets access to these treatments? It is a massive challenge. And then there's the big one, the ethical line in the sand. On one side, you have what we've been talking about, somatic editing. That's fixing the cells in one person's body to cure their disease. Pretty much everyone is on board with that.
But on the other side is something called germline editing. That's making changes to DNA that can be passed down to future generations. You're not just editing a person anymore, you're editing the human gene pool itself. And that is a whole different ballgame, and something that is, for now, strictly regulated.
And that's where we have to end it for today. The science isn't some far-off fantasy. It's arriving. The one-time cures for diseases that we used to see as a life sentence are becoming a reality. The technology works. But that leaves us with the ultimate human question. Not for the scientists, but for all of us. Now that the cures are here, who decides who gets them?